The overarching goal of my group is to develop novel magnetic resonance methods, going from molecules to mice to men on the entire translational scale. And for that we employ methods such as hyperpolarization, pulse sequence developments, and try to develop novel imaging biomarkers. And in all these cases, the imaging technologies help a lot to improve the patient's lives.
So preclinical research gives us a quite high flexibility. On the one side, we can test new contrast agents much faster, but we also can work at higher magnetic field strength. And as a last point, we have more flexibility on the scanners and acquisition protocols.
So our Bruker biospec instrument was upgraded with several hardware and software technologies. That includes novel pulse sequences that we developed on our own and implemented in the scanner. But it also includes hardware like customized radio frequency coils, especially cryo coils that at low frequencies such as carbon-13 give us an additional sensitivity enhancement. Besides high sensitivity, we also need high specificity, and there carbon-13 labeled nuclei offer us very advantages, such as for example tracking biologically relevant molecules in vivo. One prominent example would be carbon-13 labeled pyruvate and its conversion to lactate.
Carbon-13 rarely occurs in our human body, so we can look at biological processes without disturbing them. There are several processes which we can track in vivo. For example, we have pyruvate, which we can use to track metabolism through lactate or into the TCA cycle, or we can use Z-OMPD in order to look at perfusion and pH. So imaging perfusion and pH with Z-OMPD works the following. We have two carbon-13 labels at the C1 and the C5 position. C1 is not reacting to metabolism or pH at all, it thereby tracks perfusion. C5 is actually changing its NMR frequency with pH. That means it's like playing an instrument at different tones. We get unique disease information and can evaluate therapies much earlier. We have kidneys, where we can see up to 3 pH compartments, perfusion and filtration, thereby getting a fingerprint of 5 parameters within just 1 minute.
We need people from very different backgrounds, such as physicists, chemists, biologists, medical doctors, technicians, veterinarians, and they all need to work together to one common goal, which is to translate novel imaging technologies to the clinic.
So MRI software and hardware needs to be quite flexible. On the one side, we have to be able to implement new pulse sequences, and being able to visualize them and process them fast is also a key requirement. In our cooperation with Bruker, we were able to develop calibrations, sequences, and new analysis tools.
Our ultimate goal is to make a difference in patients' lives. And these changes that we detect with our imaging methods, they could lead to earlier therapy response assessment or to a clearer diagnosis, for example, of cancer lesions early on.